Good afternoon. It's good to see you all here. Some of you are faces I haven't seen before, but that's all right. So I should say 2 words about what this is. It's self-evident, I guess. We've talked about it for many years during the course of this. That's the Brooklyn Bridge. And we are the 2 people standing on the catwalk. And on one side, generally speaking, are advances in basic biological, mathematical, engineering science, and on the other side are clinicians, patients, and disease. And the whole purpose of this course and many, many activities here is to try and bridge the gap. Here, the East River, but our gap is much bigger than the East River. And there are many ways of building a bridge. This is only one. But what we've tried to do now at 17 years is bring together 3 components: a live patient who talks about their disease, a clinical investigator, a physician scientist who describes what he or she does and something about the disease for those who don't know much about it, and then a basic scientist who's really at the crest of the wave and talking about what we know, what we don't know, what we need to know. And the whole idea is to get people to speak a common language. That's the whole story. And so, okay. So the great revolutions of society, the agricultural to the industrial to the atomic, molecular, from that to the genomic to the now informatics, those have occurred at shorter intervals in the history of our society. And it's quite remarkable that today's speaker, Francis Collins, has been extremely instrumental, actually, in the occurrence, the development, and anticipations regarding the latter 2. 2 revolutions in the span of one lifetime. Who knows what's happening next? But it's been said that you can read the future only so far. And as Niels Bohr put it, prediction is very difficult, especially about the future. But we now have all sorts of tools and opportunities which are creating just worlds of amazing opportunity. But think back to the gap, because they create and add to the gap unless people can communicate together, sometimes by serendipity and others by various ventures, including something like this modest sort of thing. So I'm not going to tell you about Dr. Collins's background for 2 reasons. One, because I presume you all know. I will comment on 2 things. And the second reason is because in his talk, he's going to tell us a bit about how he got to where he is. But I would point out that Francis has been here at the NIH, initially founded the National Human Genome Research Institute at a time that ultimately led to the sequencing of the human genome and the opening up of another yet, Pandora's box. And then he became director of the NIH 8 years ago, 7 years ago. Got it. 9 years ago, excuse me. I remember once him telling me that he thought that was the most exciting job in the world. That was before he acquired it. I hope that you still feel that way. But during his tenure, amazing things have happened, including the 2 revolutions. And these are very profound. So we're very grateful to Francis for taking time to speak to us today and to delve a bit into the past, the present, and the future, despite Bohr's prediction. So today he's going to talk to us about the National Institutes of Hope. And it's a great pleasure and honor that you will do this, Francis.
Well, thanks, Wynn, for that kind introduction. And it was a little challenging when you invited me to come and give this presentation to figure out how I was supposed to be a patient and a basic scientist and a clinician. And I guess we're all patients, so maybe that one acquires simply by being alive. And I guess I have had the chance to do some basic science and continue to in my own lab right here in this building on the 5th floor. And also have had the privilege of being engaged occasionally in the translational and clinical aspects of what kinds of research have come out of that. And now with genomics finding its way into many different aspects of clinical medicine, it's not too big a stretch to say that that's going to be part of this talk as well. But it is a little different than the model that you usually bring forward. So to all of you who were expecting the usual trio, all you get today is me. Now, there is this little setup over here, and I guess Chekhov once said, when a gun appears on the wall in the first act, you can be sure someone's going to be shot by the end of the play. Well, this is our version of the gun on the wall. So by the end of the occasion, there might be some music, but not too soon. We don't want to ruin the dignity of the occasion prematurely. But Wynn, I do want to say thank you for the invitation. Even though I wasn't sure at first, it's been fun thinking about exactly how I might talk to this audience and to all the people that are watching by videocast about where things are going, because that is one of the things that I have the chance and the responsibility to spend time on, sort of realizing how fast we are moving at the present time and where that might take us in biomedical research, and how can we be sure we're doing all the right things to invest in those outcomes, particularly those that people are desperately waiting for, all those people who need that kind of sense of hope. And I do think we're not just the National Institutes of Health, we are the National Institutes of Hope for a lot of people who are still waiting for answers for the conditions that affect them or their family or their loved ones. And that is one of the reasons this is such an exciting place to be and such an exciting time, because we're right in the middle of figuring out how to offer that kind of hope. So that will be my theme, but I'm going to start off with a little bit of more autobiographical reflection, because Wynn thought that might also be appropriate, given that this is a little different than the usual demystifying medicine. Maybe I'll try to demystify the director a little bit. Again, I got to quote Wynn here about why we're here, and that cartoon, which I guess was drawn 17 years ago by Martha Blaylock, who used to be at MedArts, which now decorates the doorway as we walk in here, but depicts the basic and the clinical people running towards each other. It doesn't show you what happens when they collided. It might have been kind of a mess, but at least they were motivated to try to understand each other's language, and that is very much what Wynn has been doing over these 17 years by bringing groups together to get them to talk to each other about shared interests all the way across the most basic to the most clinical applications. And as Wynn has said, we need to do that because advances in biology and technology are occurring logarithmically, or exponentially would be another way to say it, but their application to understanding and treating human disease hasn't moved at the same pace. So creating an increasing gap. Our determined goal at NIH is to try to close that gap, and this series in particular is a critical catalyst for that. So Wynn, thank you for all you've done over these years to bring people together. And this is now a hot ticket for the speakers. You get invited by Wynn to give a demystifying medicine talk. Of course, you'd want to do that. I don't think you have too much trouble twisting arms anymore. Didn't think so. Well, I have worried about this for a while. So yes, going back, yeah, I looked a little different then, and the spectacles certainly look different. This is when I was actually writing an article that was based on a presentation I gave at one of my alma maters, Yale, about the importance of bringing basic science and clinical medicine together, and particularly warning about the risks, which are probably much worse now than they were then, about whether we are recruiting and retaining enough physician scientists who have the capabilities to go across that bridge. But of course, they need lots of help. And so in that article, certainly pointing out just how important it is that we have the benefits to society of bringing together the basic advances and the clinical applications. But let me go further back. Okay, that's pretty far back. That boy there standing next to the gate at a farm in the Shenandoah Valley with a barn behind him. Yeah, that's me. That was about age 7. I grew up on that farm. I didn't go to school until the 6th grade because my mother taught me at home, concluding that she was a better teacher than I was likely to find in the local county schools. And she was probably right, because she was a very gifted teacher. And she taught me how to just love to learn new stuff. I did manage, after we moved in town, to end up going to public school in the 6th grade, and found that science was a real call to me in a 10th grade chemistry class taught by a very gifted teacher. The other picture you see there is when I'm about 16 or 17, and yeah, I'm thinking maybe I don't want to be a scientist, I want to be a musician. Well, that's not probably a way, if you have the talent I have, which is modest but not exceptional, of making a living. So that became a hobby and not a pursuit that was going to pay the bills. By the way, when I was 7, what was I thinking there about what I want to do with my life? I can tell you, I wanted to be a truck driver. The bigger the better. Something with 18 wheels, that would be just right. I still kind of have that dream, but anyway. So yes, I went off to university then after getting excited about science, and majored in chemistry at the University of Virginia, which was my state school. The tuition was $900 a year. Can you imagine that? And I was the 4th of 4 boys in my family, which had very modest incomes, so it was up to me to figure out how to pay for that. And you could in those days by getting a decent summer job. So I majored in chemistry. I was completely disinterested in anything that had to do with biology. It seemed very messy. It is messy, actually. I was right about that. But it seemed intractable, and that's where I was wrong. It is tractable. And so I was much more interested in things that were more mathematical and digital, chemistry and physics, mathematics, and I avoided any exposure to biochemistry or biology. And finishing up my degree at UVA, decided, well, what's the next step? I better go get a doctoral degree. So off I went to Yale to get a Ph.D. in physical chemistry. And my project was entirely theoretical. I spent my days in this little space in the basement of the Klein Chemistry Lab. Yeah, I know, it looks a little grim, doesn't it? With cinder blocks next to me, and figuring out how to try to solve unsolvable equations, which is what quantum mechanics offers you, and particularly how to make predictions about what happens when you scattered protons off of hydrogen gas and transferred energy from the translational to the vibrational phase. So you can see the title of my PhD thesis, which has a lot of equations in it, which I take it off the shelf now and look at it and go, I have no idea who wrote this stuff. And along the way, I actually decided maybe I was a little too narrow, and I was in the Department of Chemistry, and there were some people doing biochemistry, and one of them, Don Crothers, was actually a pretty darn good nucleic acid chemist. And I got to know one of his graduate students because he and I were the ones who were there at 2 a.m. And he would tell me what he was doing, and it sounded pretty interesting. And I would tell him what I was doing, and he'd go, yeah, fine. So I decided I better learn something about this, took a course, a graduate course in biochemistry. And my gosh, my eyes were opened. There was this molecule called DNA. I really didn't know a thing about this. And life makes sense. And there's a digital aspect to this. And this is an information system. And of course, this was at the time where recombinant DNA was just being invented. And it was becoming clear this was not just something you could study. You might actually be able to manipulate and learn interesting things about how life works and maybe even apply it to human medicine. So this was both a revolution and a deeply disturbing sense that perhaps my planned professional path had just taken a big turn. But undaunted by that, even though at that point I was married and had a small child, decided it's time to do something different. And if I'm going to do something different, I'm going to make it really different. If I'm going to go into life science, I want to do it in a way that I can both understand the science and maybe also figure out how to apply it in a way that would be helpful to people. So what to do? I went to the University of North Carolina School of Medicine. Now here's the guy who hated to memorize stuff, and I went off to be a medical student. What's wrong with this picture? But actually I loved it because it was intensely interesting. It was about humans. It was about how our bodies work. It was about all these aspects of biochemistry that I hadn't really understood. And again, at the bottom of all of it was human genetics, which became the thing that I was pretty sure was going to be my calling without quite knowing how that was going to work. So I did my 4 years of medical school. I went on to do a residency in internal medicine. I even stayed an extra year, was a chief resident in internal medicine, and learned a little bit about how to manage groups of high-strung people, those are medical residents, and how to make sure that patients are getting good care. And then I was ready to go back to the research idea, but I didn't know how to do this. I'd never held a pipette man in my hand. I could read some of these papers on molecular biology, but I didn't really understand the methods section. So I had to do something to get grounded. And that meant finding a fellowship that would give me some additional clinical skills in medical genetics, but would mostly give me a chance to do research. I went back to Yale, a different part this time, the School of Medicine, and I had the privilege of being invited to join the laboratory of Sherm Weissman, who's still very active at this point. Sherm, who's an MD, but probably the brightest, most creative scientist I've ever had the privilege of knowing, much less working for, but also just about the worst communicator. So it was always a bit of a challenge to figure out, especially in those first few months, exactly what was Sherm asking me to do, because there were 10 ideas a day, and some of them were probably worth following up on, and I never knew which ones. And so it was a pretty tough beginning to that fellowship, and I almost quit a couple of times because I just felt like this is just not turning out to be the place where I guess I'm intended to be. But gradually things began to work, and actually began to work on hemoglobinopathies, and working with another professor at Yale, Bernie Forget, who'd been a longtime collaborator with Sherm Weissman. And my whole project then focused particularly on hereditary persistence of fetal hemoglobin, something that now turns out to be more relevant than ever as we seek ways that we might ultimately cure sickle cell disease, which I might just come to in a bit. So after 3 years of that, it was time to get a real job. And you might ask, how old were you at this point? Well, all right, I was 34. And looking around to see where was an academic institution that would allow this kind of flourishing that I hoped to do, I ended up landing at the University of Michigan, which was very good fortune because the chairman there, Bill Kelly, was determined to create an atmosphere for young assistant professors who wanted to do research where their time was really protected. And he also had a bit of a war chest, and he would allow you to buy equipment that you didn't currently have a grant for if you thought it was going to be worthwhile to enhance your own potential for making progress. And he also encouraged us to tackle projects that were high risk and not just do the next obvious thing. And as an assistant professor just starting out, when you're intensely anxious about whether you're making it or not, it's tempting to do the next obvious thing and try to get some papers published. But maybe that's not the right answer. Maybe this is the time where you're at the best place you'll ever be to tackle a really hard, really important problem and throw everything you can into it, working yourself at the bench with other people next to you. And Bill Kelly made that kind of opportunity a reality for me. And so that's what I did. And the goal at that point was to try to figure out how can we take all these developments in recombinant DNA and understand what really are the causes of thousands of genetic diseases that we know are inherited in dominant ways or recessive or X-linked ways, but we have no idea what the gene is that's involved in those diseases. We don't know enough about the protein biochemistry to predict what the actual gene is. We just know it's inherited. And that may seem like a trivial thing to ask because now, today, that's a problem that most of you could solve if you had access to a thermal cycler and some family DNA samples and a DNA sequencer in the space of a couple of weeks. But back in the 1980s, there was no Genome Project. You couldn't click your mouse on the genome and see what was there. There were very clunky, rudimentary maps of chromosomes. They were getting a little better as time went on. There were genetic markers that were sort of displayed across all of those chromosomes. And if you wanted to try to map a particular human genetic disease, with great trouble, you could do so using this strategy of linkage that had been introduced for human genetics in about 1980 by Botstein and White and Davis and Skolnick, and had been shown to work for Huntington's disease in 1983. But it was incredibly painful. But this seemed like a worthwhile thing to do for somebody who's a physician, who is interested in genetics and kind of excited about the technology. So I set up my lab to try to do those things and started working on cystic fibrosis as a disease that desperately needed new answers, the most common potentially fatal autosomal recessive disease of people with Northern European background, about which in 1985, when we started working on this, really nothing much known about the physiological defect. We knew there were problems with infections and thick mucus in the lungs and in the pancreas. We knew that chloride was elevated in sweat. So there was something going on with ion channels, but nobody really quite knew what they were looking for to go hunt for this gene. My lab developed a means, if you could find a piece of DNA that was somewhere near the gene, you could start jumping towards it, as opposed to what was available at that point, which was walking painfully towards it. And if you had to cross maybe megabase or more, walking was not going to get you there in less than a decade. So chromosome jumping was something that I'd started working on as a postdoc and then was a big focus of the lab when I got to Michigan. But it was tough. I thought of myself as a gene hunter. People kept saying, well, you're hunting, but why haven't you found anything? And that required, at one instance, the need to go to a barn nearby the university and have my picture taken, sitting in a haystack, holding up a needle, saying, people, this is what it's like, trying to find the cause of cystic fibrosis. Yes, that is a real chicken. And actually, it was even harder than that when you started doing the math. Finding a single nucleotide out of 3 billion is a lot harder than a needle in a haystack. So yes, that was maybe a way of apologizing for not making it happen quickly. But ultimately, thanks to the hard labor of a remarkable, talented group of individuals that I had the privilege of mentoring in my lab, and also especially linking up with the laboratory of Lap Chi Choi at the Hospital for Sick Kids in Toronto, because we were both trying to find the cystic fibrosis gene, and we had complementary approaches, and we'd been competing. And in 1987, we met and said, this isn't any fun. Let's just do this together. And so his lab and my lab essentially merged across a fairly long drive across southern Canada, and we covered that road many times, going back and forth. And ultimately, over the next 2-year period, it worked. And we found just a 3-letter deletion of a CTT in the middle of a gene whose identity had never previously been known. It was in territory that nobody had mapped. A gene that once you had its exons mapped out, you could tell really did look like it was an ion channel, but sort of a different one. It was an ABC transporter, which is a family that contains many other famous genes, and nobody knew that CFTR would be one of them. And that's what we called it, CFTR, for the cystic fibrosis transmembrane regulator, which said more than we actually knew, but we had to call it something. And that mutation, delta F508, turns out to be the common one. Cystic fibrosis would not be so well known, except that that particular mutation has a high allele frequency in Northern Europeans. We still don't know whether that's because it's been selected for in heterozygotes or whether it's just drift. At any rate, that was a big moment, because that was the first time that a gene for a well-behaved Mendelian disorder had been discovered without any evidence and the protein or the physiology level of what you were looking for, and without any cytogenetic rearrangements to help you either. Others had been found because of chromosome translocations that interrupted the gene, and those were nice neon signs to tell you where to look. Cystic fibrosis didn't offer any of that. And that, I think, encouraged a number of other projects, including our own. The Huntington's disease project, which I had gotten engaged in also several years before, and involved a collaboration of 9 laboratories over time, ultimately found the cause of Huntington's disease in 1993. Neurofibromatosis, the most common disease that I, as an adult medical geneticist, see in the clinic, had its gene discovered by my group in 1990. Turned out to be a very interesting gene that's involved in the RAS pathway. And more recently, as the lab has continued that effort, And since I've come to NIH, discovered a few other such causes. Maybe most dramatically, for me anyway, has been this discovery of the gene for the most dramatic form of premature aging, Hutchinson-Gilford progeria syndrome, which causes cells and the individuals who have those cells in their bodies to senesce rapidly. So the average survival is only about age 12. And you can see Meg there in the big hat was the first patient I ever met with progeria when I was a fellow at Yale. And then Sam Burns, the boy between me and Cookie Monster, who became a dear friend and who died just about a year and a half ago. All of these kids are incredibly inspiring because their intellectual development is, if anything, precocious, and yet their bodies are aging at this prodigious rate, and their future is pretty grim unless we come up with something to help them. Happy to say that just last week, first paper published showing that one of the therapeutics that we came up with after knowing what the defect was has shown actual prolonged survival for kids with this disease. Not as much as I would hope, and we have another potential intervention using morpholinos that may be even more promising, but has not yet gotten approval for a phase 1 trial. We hope to get there within the next year. So all of those things were exciting detective stories, but detective stories that had a big human significance. But going back to where we were with cystic fibrosis, the idea that you could actually imagine doing this for those thousands of diseases that we knew were inherited in well-behaved ways, but without having any idea where you were in this sea of DNA called the human genome, it just seemed absolutely impossible. And so I was one of those who was particularly pleased that discussions began happening about that same time about organizing an effort to actually determine the complete sequence of the human genome and not have this done in a bit-by-bit cottage industry way, but actually organize the whole thing and do the whole thing in an efficient fashion. And so various reports came out about this and hearings were held. And here at NIH, the Genome Project was officially launched in October of 1990 with the director, none other than Jim Watson himself, the same Watson who with Francis Crick had uncovered the double helical structure of DNA with a little help from Rosalind Franklin, whose data they looked at without her permission. I have to mention that. But here was Jim, who's probably the rock star of biology, willing to step forward and lead this enterprise, which got a lot of credibility in the scientific community and on Capitol Hill. And so the project did, in fact, get underway. And I was delighted. I applied for and got one of the first Genome Center grants at the University of Michigan, began working away on how we were going to actually do this incredibly challenging task. Because think about this. In 1990, when this started, people were publishing papers in fairly reputable journals where the main guts of the paper was that they had sequenced a few hundred base pairs. It was like, that's a publishable entity, and we're going to go for $3 billion and the technology to do that. Have you invented that yet? Well, no, but somehow it will happen. And Jim had promised we would do it. We would do it in 15 years. So we all had to be held accountable, and we were willing to because it seemed like it was a worthy goal. And either we would succeed or at least we would do our best and try. You may not also know that the Genome Project at that point was intensely controversial. That probably half or maybe more than half of the scientific community was opposed to it, arguing that this was unfeasible. It was just not going to be possible to do this. So you're making a promise that was going to fail. Arguing also that it was going to take money from other things, and the estimates of what it was going to cost would probably balloon out of control, and the rest of science would suffer. And then most offensively to people like me who were interested in working on this, that only mediocre scientists would want to work on this because it was going to be so mind-numbingly boring just reading out all those letters of the DNA code without actually attaching it to a particular biological or medical question. Fortunately, those arguments did not carry the day. In particular, leaders like David Botstein and Maynard Olson, who had been deeply skeptical, became convinced that this really was a critical effort and made their case rather compellingly. So here we were. The project was going. At that point, Bernadine Healy was the director of the NIH. Jim Watson, as the director of the Genome Project, therefore reported to her. Jim Watson is not somebody who reports easily to anyone. If you know him at all, as I do, Jim is somebody who says what he thinks and doesn't wait too long to worry about what it might imply. And he and Bernadine got into a tussle, specifically about whether or not it was right to file intellectual property claims on DNA sequences, which Jim felt was the wrong thing to do. And Healy, as the NIH director, said, well, maybe it's the wrong thing to do, but I wouldn't be holding up the public trust if I didn't engage in this, because it might turn out that this would result in a return of resources to medical research, which would be a good thing. And after a couple of disagreements spilled out into the public, and Jim used the term lunatic to describe his boss, not once but twice, he was gone. And then she was on the phone, and she was calling me and saying, I think we need a new director for the Genome project, and I hear that you might be a good person to do this. I'm not quite sure why she thought that. This was certainly a surprise to me. And I was really happy. I was in Ann Arbor. I was taking care of patients. I was teaching medical students, which I enjoyed doing, and I was mostly running a research lab that was going really well. And the idea of packing up all of that and coming to become the one thing my mother told me never to be, a federal employee, just didn't seem like a good idea. So I said, no, it's not me. Well, she called back. And over the course of 6 months, ultimately, I began to realize, what, am I crazy? This is probably the most historic, critical project that's going to happen in life science in my lifetime. Yeah, it is potentially doomed to fail, but it might actually succeed. And it would be an amazing thing to try to bring together some of the best and brightest minds of the generation to work on it. And I think it is pretty compelling, and I bet I can get some of them to join on. Some already had. So, okay, against my mother's judgment, it's probably time to say yes. So I picked up, brought my lab here, brought some other distinguished human and medical geneticists here from all over the country to start the intramural program. There had been no intramural program for the Genome Center. It was a center at that point. It was purely extramural. And then it started as an intramural program when I arrived in 1993 over in Building 49 at the time, and still a bunch of genome folks over there. And gradually over time, we've expanded to where we are now, including folks here in Building 50 and people up in Rockville at NISC and a few other places as well, up in Baltimore, in fact. So that was unexpected, but it did then plunge me into a very different role for which I was totally unprepared. I'd never managed anything bigger than my lab, and now suddenly I'm supposed to manage this project, which involved 20 laboratories in 6 countries, all of whom needed to work together, needed to agree to common standards and timetables, and some of whom actually weren't doing very well, and we had to figure out what to do about that. But over the course of the next 10 years, from 1993 to 2003, I had the great privilege of having a chance to nurture this enterprise, to recruit some amazing scientists to work on it, who all agreed to put their shoulders to a common wheel and not worry too much about who got the credit. And that led to a series of milestones over time, initially with model organisms, with genomes that were a bit more tractable, starting with simpler things like yeast and working up ultimately to the human. And the real human sequencing got underway in about 1996 with a absolutely critical decision made at that point that all the data would be made immediately available. Every 24 hours, we would post the results of that day's work so that this would be accessible to anybody who had a potential use for it. And once it's in the database, you can't claim it for intellectual property because it's already in the public domain. That was a very intentional effort, and I got to give a lot of credit to the late John Sulston, who pushed for that particularly strongly, my wonderful colleague from the Sanger Institute in the UK. And it was an intense experience, and I still, when it's 11 o'clock in the morning on Friday, I get this little slight anxiety about, oh boy, are we ready for the conference call? Because every Friday at 11 o'clock, we would have the principal investigators of these centers on the phone, and we would have a very significant accounting of what people had accomplished, and also try to turn it into something interesting and have it be a bit of a lab meeting as well. Things got underway. The scale up of sequencing got better. By 1998, we could actually see we were going to succeed, but we still had a lot of work to do. We had a billion base pair bash at the National Academy when we sequenced the billionth base pair. That was close to being accurate. And then we kept going. And in the year 2000, the picture you see there with Bill Clinton, that was June 26, 2000, when we announced that the draft of the human genome sequence had been achieved, announced this jointly with Craig Venter and his company Celera, who had been carrying out similar activities, although because all our data was public, they were using their data plus our data, which is only fair. But at any rate, we decided at this point, both entities have done pretty well. Let's declare joint victory, and we did. Shortly after that, Celera stepped out of the business of doing DNA sequencing because it was pretty clear there wasn't a business plan here if the data was going to be immediately accessible and free to everybody. It took another 3 years after that to finish the job. A draft is fine, but any of you who've worked on hard projects, you know the last 10% is at least half the work, and it was. But by 2003, we had covered all of those missing pieces, except for a few things like centromeres that are still somewhat outside of our grasp. People make fun of the fact that we've finished the human genome about 6 times now, but and we're probably not done yet. But the main claim was 2003. And that picture you see there is just of some of the leaders of that effort, 2,500 scientists, 20 institutions, 6 countries, 13 years. Now, 13 years means we finished in 2003. Watson had said 2005, so we were 2 years ahead of schedule. The budget that had been laid out for this and approved by the Congress, I'm happy to say we didn't need all of it. We ended up spending $400 million less than what was expected, and that helped me a lot going forward, and still helps me to say it is possible for the government to manage a project that is ahead of schedule and under budget. Now, of course, that was a great moment, and I would not trade anything for the experience of basically seeing this emerge, but we had to be honest with ourselves. We had no idea how to read this genome sequence. There it was, and you could use some algorithms to find the exons, and that was like 2%. And the rest of it, well, it's probably important, but it's pretty hard to know what it's doing. So it became critical right after this, or even before, to begin to map out other large-scale projects. We needed to understand variation in the genome. We had this reference sequence, which was actually a mosaic of donated DNA from 6 people whose identity we still don't know, because that was the intention. But we needed to understand variation in a much broader scale. And we need to understand gene regulation and what are the regulatory signals and how do they influence which genes are turned on in which tissue. But I got, at that point, after running a bunch of those projects, ready to move on. And so by 2008, after a lot of those things had happened, I decided it was time to take a break. And so they gave me a nice party. There was a wonderful band that played, and I got to play with them. And they took up a collection and made it possible for me to design my own guitar. That happens to be right there. You will notice the double helix on the fretboard in Mother of Pearl. And of course, every guitar has to have a name, and this is Rosalind, as in Rosalind Franklin. And I got to spend more time with my wife and more time riding my motorcycle. And I thought, boy, this is pretty nice. And I wrote a book about personalized medicine. And then I got a different call about public service. And this was from a president. And so back I came to NIH in 2009 to serve President Obama as his NIH director. And yeah, Wynn said at the beginning, I had once said, this is the best job in science. Do I still feel that way? Yes, I do. I felt that way walking in that first day, and I feel that way today, because the landscape of opportunities that are right now in front of us is just breathtaking. And to have a chance to be part of that and to identify something that looks like it's bursting with potential, but maybe with a little more push, a little more encouragement, I could get there a little quicker. That is really a joy. And to work with the amazing people who are here at NIH, those 27 institute and center directors, the scientific directors, many of the principal investigators in the intramural program. This is an amazing place at an amazing time, no doubt about it. So maybe I should then say a little bit more about what I think NIH is able to do in the frame of this trying to predict where we've been and where we're going. Again, the mission of NIH seems a lot like the mission of Demystifying Medicine. It's to bring together these two parts of what we do, pursuing fundamental knowledge about the nature and behavior of living systems, but figuring out how to apply that. Isn't that what you try to do, Wynn? Yeah, bring those together. The history here is a long and distinguished one. Don't need to dwell on it too long, but goodness, NIH has been around since 1887, but we weren't here then. We were in Staten Island, and then gradually moved to DC, and then ultimately out here. As you can see from the photograph, a rather famous moment when Franklin Delano Roosevelt, as the president, dedicating from the steps of Building 1, I walk up those steps every day, the grounds of the Bethesda campus. And over the course of time, beginning in 1948, went from being the singular National Institute to becoming plural, very plural indeed now, with 27 institutes and centers, an organizational structure that only the Congress could design, but which actually works really well because of the remarkable leaders that head up all of those institutes and who are very determined to figure out ways to be collaborative. I don't think we have silos around here, at least not anymore. And of course, NIH both does research in its own laboratories, but supports that across the country. Those of you in the intramural program maybe haven't thought so much about that, but that's where most of our money goes. And we do a lot of training, and we foster communication and medical information. And it works pretty well. Latest tally of Nobel laureates, 153. That's not bad. And I predict come next October, that number will go up because every year it does. And the clinical center here as a remarkable center of the clinical activities of the world, but certainly here in the Bethesda campus, sort of a jewel of our enterprise. Initially built in 1953 when it opened. I remember the first time I came to NIH, I was applying to medical school. I was finishing up my Ph.D. at Yale in quantum mechanics, and I had read some papers by Ira Paston. And I thought they were pretty interesting. And I wrote to Ira Paston saying, I'd like to drop by your lab, because I was coming to do a interview at Hopkins, and I thought that couldn't be too far away. And maybe we could talk about my idea, which I'm sure was incredibly naive. And Ira wrote back saying, well, we'd love to have you. Why don't you come on this day, and we'll convene some of the other people in the lab, and we can talk about your idea, which I had sent him in a little 2-page thing. And so I did. And he had his group of 5 or 6 people and talked about the proposal that I had come up with, which was purely theoretical, because remember, I was a theoretical chemist. And they gave me some good ideas, and they were incredibly generous. And Ira was the most generous of all. What an amazing person, and as he still is, an amazing person. And then I left, and I started to walk across the NIH campus, and I walked past that building, Building 10. I said, that's the biggest building I've ever seen in my life. What goes on there? This is a really fascinating place. I'll have to learn more about this someday. Remember that very clearly. What was I? This was 1972. Yeah. Okay, well, it is an amazing place. And of course, it's gotten even bigger and more remarkable with our new clinical research center and all the things that go on there, 1,500 clinical studies, winning the Lasker Bloomberg Public Service Award, being depicted, and I hope all of you saw this in Discovery's special called First in Human, that 3-part series talking about patients who come here for clinical trials and the remarkable way that this intersects between clinical medicine and basic science. Just an amazing place. And the impact that we have had between intramural and extramural in terms of life expectancy and health span of all of us is truly amazing when you look at it. Here's just an example over the last 35 years or so, 45 years, I guess. But you can see cardiovascular death rates having dropped more than 70%, cancer death rates now falling more than 1% per year, HIV, which was a death sentence, now associated with almost a normal life expectancy. And I could go on and on. So all of you who are part of this, you should feel really good about the way in which this institution and the scientists who work within it and who are supported by it out there and all those other places across the country and across the world have really changed things in a dramatic way in the last few decades. But now, where is it going? I took your Niels Bohr quote with some alarm because I'm about to do what Niels said is really hard. Actually, I thought that was Yogi Berra who said it, but maybe he stole it from Niels Bohr. Prediction is risky, especially about the future. Well, okay, we're going to try and see if we can travel into the future. Probably most of the people here who are under 35 have no idea what that vehicle is, but it played a significant role in a particular Back to the Future movie. That's a DeLorean. So let's get in the DeLorean and see where we think we might be able to go. And I'm going to pick somewhat on my own sort of personal preferences, 10 areas that I think in the next 10 years are going to be dramatic in terms of the advances that will happen. And if I don't pick the area you're working in, please don't take that personally, because I could readily have picked many more than 10, but then we'd be here much too long. So I'm just going to pick these, and I think I can convince you that all 10 of those are pretty exciting, but no doubt there are many others as well. So a little caveat there. I don't want to get any nasty emails later on today. So let's start. And maybe we'll start with something that's a bit more in the basic science arena. 10 medical advances in 10 years. Single cell analysis. I got to say, you could argue that there have been maybe 3 major advances in understanding life. One would be evolution, that nothing in biology makes sense except in the light of evolution. That's Dobzhansky's quote, and it's true. Another would be the DNA to RNA to protein, the informatics, the flow of information that we understand is how living things control their functions and pass their information on to their offspring. But the third one is basically the fact that living things are made up of cells. We take that for granted because it's so obvious, it wasn't always known, but that is the unit of life. Regardless of what organism you're talking about, okay, viruses are over here, but most of the time we're talking about cells, and yet we have not, until very recently, had the ability to look at a single cell and ask, what's it doing? And now we can. And that seems pretty profound. Certainly we've done this by having cells growing in a Petri dish that are clonal. So when you do an analysis on 10 million of those cells, you kind of can infer that that's what a single cell would have looked like as well. But there's a lot of interesting questions in biology that you don't want to just look at clonal cells growing in a lab culture. You want to look at tissues in vivo. And as soon as you start doing that, if you have to grind up a piece of tissue, you know you're diluting out and blurring out an awful lot of the biology. My lab works on diabetes up there on the 5th floor. We're really interested in the pancreatic islet. And we can get pancreatic islets from humans sent to us as part of the distribution system, and we can make RNA from those. And we've done that a lot and do RNA-seq and see what genes are on. But we know that we're looking at a mix of cell outputs from alpha cells and beta cells and delta cells, and probably a whole bunch of other things that haven't even really been well recognized because they're such minor components. So we're missing out on a lot of the biology. Well, not anymore. We're not doing that anymore. Now we're basically disaggregating those islets, sorting them out into single cells, and doing RNA-seq or ATAC-seq if we're interested in the chromatin, or DNA methylation if we're interested in the DNA methylation epigenomics. This is now possible. And I suspect many of you are also in this space as well because it's come along pretty quickly. Among the things that are happening here is the Human Biomolecular Atlas Program, HubMap, which is an international effort that we're part of along with the Wellcome Trust and along with the Chan Zuckerberg Institute, because Mark Zuckerberg and Priscilla Chan have gotten very interested in this space as well. So we're going to learn a lot about individual human cells just as a database, a reference database in a few years. If you're interested in knowing, well, what's really going on in the liver, or what's really going on in the pancreas, or in some other tissue, you'll be able to have that as a reference set, knowing what cells are making which transcripts. And of course, this also provides all kinds of opportunities for medical applications that we didn't have before. One of the projects that I spent a lot of time on is building something called the Accelerating Medicines Partnership, which is where NIH is working with industry to try to identify new targets for common diseases. And one of those projects is on rheumatoid arthritis. And we know in rheumatoid arthritis, there's a lot of things that are happening in the synovium, in joints that are being eroded. And we know the immune system is overactive in that space. But if you take synovial tissue and look at RNA expression, you get a mishmash. It's very hard to interpret. Well, now we don't do that anymore. So this is synovial biopsy that was first sorted into CD45 positive cells, leukocytes, and then CD45 negative stromal cells. And then individual RNA-seq carried out on each one of those cells. Each dot you see there is an individual cell. And then take that RNA-seq data and using k-means clustering, turn this into a two-dimensional plot. And you can begin to see there's a lot of structure here. There's a lot of interesting findings, both in the leukocytes and in the stromal cells, various subsets that we didn't know were there before. This is just one small example of things that are happening all around us because we can now look at single cells. This is going to really open up. I mean, whatever you're working on, imagine that if we had the full capabilities of doing this for those tissues, it is going to open our eyes. And again, it's coming back to this fundamental thing. Life is made up of cells. We can now look at them. We don't have to guess what was there. We can look at them. People are even proposing protocols where you could, with a single cell, look at RNA, look at chromatin, look at methylation, all in one experiment. I think that's entirely doable in one cell. And of course, you would already know the DNA genotype because that's going to be more stable in a bunch of cells. So this is moving us in interesting directions, and it will increase our understanding of lots of disorders, including, you can see here an X-ray of somebody with really bad rheumatoid arthritis whose joints are being eroded by those cells that I just showed you the map of, and now we have a better idea about who's there and what are they doing. And of course, number two, I've got to talk about the brain, because this is a moment where we're just brave and bold enough to say we think we can understand how those 86 billion neurons between your ears do what they do. This is arguably the most complicated structure in the known universe, and it's right here. And it's incredibly efficient in terms of its power demands. And each one of those neurons has about 1,000 connections. So the complexities here are truly daunting. And you might wonder, why are we so bold to say we can do it? Well, a lot of it is because of development of technologies that allow us to begin to see not just the whole brain at a time or one cell at a time, but the stuff in between, the circuits. This is a really nice movie from Carl Dieseroth. This is a mouse brain that's been subjected to his technology called Clarity, which basically takes the brain and puts it in an appropriate bath so that all the lipids are leached out and it becomes optically transparent. And so now you can take a little tour through it, which I hope doesn't make you nauseated because it's a little jarring. Oh my, where did we go there? And you're now looking through, and this is done in a way so only some of the neurons are stained, otherwise it would be too crowded, but you can begin to see in 3 dimensions the kind of information we've never really had before. And even after Clarity, the brain is still totally susceptible to all kinds of immunohistochemistry, so whatever particular circuit or cell type you're interested in, you can stain for. And there's no reason you can't apply this to even larger structures like the human brain. In fact, that is being done. Giving you, I mean, you can see individual cell bodies there, the kind of 3-dimensional perspective that we've really lacked before, but now are getting access to. And it is a very complicated structure, to be sure. And when I talked about single cells, well, we need to be able to look at those too. This is a tiny segment of a mouse brain to just give you a sense of how complicated this is. And they basically, by electron microscopy here, figured out everything that's in this little segment, and they sort of blowing it up here so you can see visually what was contained within that little chunk. And in a minute here, they will basically then define which kinds of cells are which by their properties, and you'll begin to see what's there. So here it's put all back together, and now it's taken apart again, and you'll see, okay, different cells, axons, glia, dendrites, but each of those also have subsets. All of that was there in that little piece. Now, are we crazy to think we might figure that out for the whole brain? I think the technologies might just get us there, maybe even in 10 years. So we have an initiative called the Brain Initiative, which is international, but NIH has at the moment the largest investment in it. And most of it has been inventing new technologies, and most of our grantees have actually been engineers figuring out ways to see what's happening in real time with circuits, initially with model organisms, but aiming to do this for the human. So 10 years, watch neuroscience. It's going to be a wild ride. And once you have that foundation of understanding normal function, think how much better off we'll be trying to understand things like autism or schizophrenia or Parkinson's, where you can see, okay, that was normal. This is where things have gone wrong. Oh, that's what we should do to fix it. Along with that, now my prediction number 3, we desperately need, and so I hope I'm right about this, that in the next 10 years, we will get them successful targeted therapies for Alzheimer's disease, because this is a condition that now already affects about 5 million people. The cost of this is approaching $300 billion a year. I said billion. And it will be $1 trillion by 2050 if we don't come up with some way to prevent or at least slow down the onset. One way is by trying to intervene as early as possible. What you're looking at here are PET scans of amyloid in the brains of individuals who are in their 30s. And these happen to be from a family that has an inherited mutation in the presenilin-1 gene, which carries a very high risk, 100% essentially. If you have the mutation, you're going to get Alzheimer's disease in your late 40s. None of these individuals have symptoms yet, but you can see from the scans, the gene carriers have a lot of amyloid already, and that is in fact the case with late onset Alzheimer's as well. By the time somebody starts to show cognitive decline, they will have been depositing amyloid for 20 years. So we can now identify who those people are, and they're the ones really that we are now focusing most of our therapeutic interventions on. If you wait until somebody has cognitive symptoms, they've already lost a significant fraction of the neurons. It's very unlikely you're going to bring those back. So the idea is to move early as possible to identify the risk, then intervene. And there are some good ideas about how to do so. Admittedly, it's been a pretty tough road here. There have been hundreds of failed trials, but most of them have been tried on patients for which it was probably too late. Imagine if you were trying to test whether statins worked by giving them to people with congestive heart failure. You probably wouldn't be very impressed. Same idea here. You've got to start early. And we also need better ideas about targets. Amyloid is not the whole story. Tau is not the whole story. There's a lot of other things going on in the brain. So these ideas then of taking a systems biology approach to what's happening in the Alzheimer's brain is also beginning to yield fruit. And we have out of that a number of new ideas about drug targets that industry is very interested in investing in. Okay, number 4, gene therapy. Rare diseases caused by single gene mutations are one of the areas where we desperately need more answers and more successful interventions. And yet gene therapy has gone through a pretty difficult period since the early period in 1984 or so when we were starting to try this. And there was always this promise, oh, it's going to work, and then it didn't. Or in the worst case, it actually did harm, as in the case of Jesse Gelsinger back at the turn of the century. But the time has now started to arrive where gene therapy, particularly using vectors like AAV, is turning out to be pretty dramatically successful in some specific applications. And I want to show you just one example, which is spinal muscular atrophy. This is a heartbreaking disease, relatively common, autosomal recessive. These babies appear physically normal at birth, but their muscle tone is poor, and it gets worse and worse. They're the floppy baby syndrome. And because the problem is in the anterior horn cells of the spinal cord, this is kind of like Lou Gehrig's disease, except in an infantile genetic version. And these babies will never learn to sit up on their own, and they will invariably die by age 15 months, basically of suffocation because they can't breathe. And one of the more heartbreaking conditions you will ever come in contact with. Well, we know the molecular defect. It's a mutation in the gene SMN1. And so it would seem feasible that you might be able to treat this if you had a very efficient way of delivering a normal copy of that gene to the anterior horn cells of the spinal cord. Oh boy, that sounds pretty hard, and it is hard. But working with a variety of different versions of AAV, Jim Wilson came up with one which is called AAV9, which does seem to get across the blood-brain barrier. And Jerry Mendel at the Nationwide Children's Hospital in Columbus, who has a wonderful GMP facility then for making large titers of AAV, decided to give this a try. Now this is infused intravenously, and you're counting on a large dose of this virus to actually get through the bloodstream, across the blood-brain barrier to the anterior horn cells in these babies, and to actually effectively take the place of that missing gene. And guess what? It works. Have a look at Matteo. He's 2.5 now. He was born with spinal muscular atrophy. Look at him up on his tiptoes, pushing that elevator button, and now he's going off to work. And here he is with his dad hanging on the monkey bars. If that doesn't inspire you, I don't know what would. That's unbelievable progress in a previously uniformly fatal disease. And they have now treated 12 kids with this, and every one of them has had benefit. The ones that have had the greatest benefit are the ones where the treatment was done at the earliest point. Matteo was treated at just 8 weeks. He'd had a previous sibling who had SMA, who died, and so his parents knew they were at risk, and so they knew right away by testing that he was affected and got him into the protocol. And now, having seen this, we need to start testing all newborns for SMA because we've got a promising treatment. That makes you feel like something's happening here, and it's not just in this one condition I'm telling you about. It's happening in a variety of other places. Hemophilia, especially hemophilia B, we're seeing what looks like remarkable advances. And it's been hard fought. So we're going to see a lot more of that in the next 10 years. You kind of get the feeling after a lot of struggle, we're on an upward curve. And then there's CRISPR-Cas, where not just by inserting a viral vector that's carrying the missing gene, you can actually go and fix the thing that is misspelled using the gene editing tool called CRISPR-Cas. And I'm going to be bold enough to predict that it won't take 10 years to cure sickle cell disease. I think we'll do that in the next 5 years. I was just at a meeting where the CEOs of the biotech companies that are doing gene editing, so that's CRISPR Therapeutics and a few others, we're all there, and at least 2 of them are now planning to start a clinical trial this year. The idea is this is a disease of the blood system, so you take the bone marrow of an individual with sickle cell disease, you sort for the stem cells, you apply CRISPR-Cas either to directly fix the sickle mutation or somewhat easier to turn on fetal hemoglobin production, something which harkens back to what I did as a postdoc. And then you expand those stem cells, so you got a lot of them. You need to do something to make space for them, so you need to do at least a limited ablation, that's the risky part, of the existing bone marrow, and then you infuse back the cells that have been fixed. I can't see why that won't work. And it might work right here with John Tisdale and the group that has been doing bone marrow transplants for sickle cell disease and is therefore very experienced in knowing just what it takes to try to manage this kind of intervention with patients who've had a lot of organ damage as a result of the disease. But if you could do it for sickle cell disease, you sure wouldn't want to stop there. And one of the things which I think is going to be really interesting is could we figure out how to deliver the gene editing apparatus of Cas9 or something like Cas9, along with a guide RNA that tells it where to go and make that change, and do that in vivo. Because a lot of the genetic diseases you'd want to treat, you can't take the tissue out and treat it and put it back, because a lot of them affect the brain, for instance. So we have just funded, through the Common Fund, a $190 million program to encourage people to come up with creative ways to do that delivery of the gene editing system in vivo to an appropriate tissue to see whether we could start to really scale this approach to those thousands of diseases that are still waiting for answers. And I think that could work. And you're wondering why I haven't talked about the stem cell revolution. Well, here I'm going to, because this is another remarkable advance that we're just beginning to appreciate where it can take us. It's only 10 years since Shinya Yamanaka in that paper in Cell that gave me chills when I read it. How many people have read a paper that gave you chills? Doesn't happen all the time, does it? This one did, where he figured out if you just add those 4 genes to a skin cell, you can encourage it to go back in time and become pluripotent, what we now call an iPS cell. And that if you then develop the appropriate cocktails of growth factors, you can turn that into almost anything. Again, my lab upstairs, using this now to try to understand diabetes. We have iPS cells that have been generated from 50 patients who have either diabetes or normal controls, and we can now differentiate those into beta cells. And these are patients whose phenotypes we know extremely well because we've studied them for 20 years. But now we have a cellular phenotype from their insulin-creating cells to put together with what we know about their in vivo findings. And that opens up all kinds of possibilities. And that's just the research side. Obviously, there are other things we can do. The National Center for Advancing Translational Sciences has been leading an effort to basically create biochips that have on them little organoids made from cells that started out as iPS cells and then got differentiated into heart or bone or liver or brain or kidney, and even to put them on a chip where there is some communication between them. So this is starting to sound a little bit like a human system, but yet you can do this from anybody. You could do it from you. Would you like to have you on a chip? This is possible to imagine. And this is a great way to test for drug toxicity. It's a great way to test for efficacy of a new therapeutic if you load the chips up with the right kind of cells. But ultimately, it's also a step towards figuring out how we might make it possible for everybody to be their own organ donor if you need that. If you need a new heart, you need a new kidney, well, make your own. There's no reason I can see why, with a combination of using the iPS cell approach, the appropriate kind of engineering of tissues involving scaffolds, probably involving 3D printing of tissues, which we now are learning how to do as well at NCATS and in other places, that would not happen in the next 10 years. And wouldn't that be awesome, given the shortage right now of organs for people who desperately need them? Maybe we could do something about that that would be pretty profound. That was number 6. Oh yes, you want a biochip. Number 7. Okay, let's figure out what is the greatest threat to current society across the world in terms of survival. It is probably not bioterrorism caused by human. It's bioterrorism caused by nature, and it's probably the next influenza pandemic. This is the 100th anniversary of 1918, when at least 50 million people died from that influenza pandemic that swept the world. And we are not out of the woods from that happening again. And as you know, our current approach to influenza requires guessing each year what strain is going to be prevalent, and then building a vaccine against that and crossing your fingers and hoping it worked. But if a pandemic strain came out of Asia or somewhere else, that particular flu vaccine that we prepared for this year would probably be of no use. And it takes way too long to develop a new vaccine for an unanticipated pandemic. Could we do better than that? Why is this so hard? We've been doing it the same way, using eggs, for heaven's sake, for a long time. Well, maybe there's a way around this. So that's the coat of the influenza virus, and that's the hemagglutinin protein, which is the major antigen that your immune system responds to. And your immune system kind of looks at what's closest to it, which is the head of that hemagglutinin, but there's a stem under there. Now, if you look at multiple influenza strains over many, many decades, what you see is that the head, as shown in red, has all kinds of variations that happen year after year. So nothing about the head stays very much the same. But the stem kind of has to stay constant, otherwise the whole thing falls apart. So if you could convince your immune system to ignore the head and instead to make antibodies to that invariant stem, and particularly that yellow area, which seems to be the same for all flu viruses, then wouldn't you have something pretty useful? Wouldn't that then give you protection, not against this year's flu or next year's flu, but all of those, including the next pandemic that we don't even know about yet, because it'll probably have that same stem. That's what Tony Fauci and his team are now ratcheting up in terms of an intense focus on getting there. And it's not trivial to convince your immune system to look at the right thing. But we're already in a phase 2 trial, just started last week, of one of the approaches to that, where you have to generate just the right structure for the immune system to look at. So in 10 years, I think we'll succeed at this. I just hope we have enough time to get ahead of whatever that next really big pandemic is going to look like. And so, yes, maybe we'll have this vaccine protecting against all strains. I'm getting close to the end, but 8, I have to reflect on our current national crisis. And that is the opioid crisis, which is all around us. And I suspect you've all been reading about it and hearing about it, and maybe even have family or friends who are engaged in tragic ways with the way in which the overprescription of opioids, which began in earnest in the late 1990s, in the misunderstanding that somehow that would not lead to addiction for people who actually had pain, and that was dead wrong, has now resulted in about 2 million people who are currently addicted. And many of them, 80% of them, starting with prescriptions. And then as their needs grew greater, and as their brains, which get rewired by this experience, become utterly insatiable to the point where just willpower is insufficient to give anybody protection against the craving that is going to drive your life and drive you to make really dangerous decisions, most of those people ultimately end up seeking out heroin. And the heroin now that you can get is all too often laced with fentanyl, which is about 100 times more potent, and which, if there's a little too much in that particular packet that you just bought, is going to kill you. And which last year killed more than 42,000 people. And this year is probably going to be higher than that. So we have a desperately difficult situation. And it was not sufficient to say, well, we just got to figure out how to stop all the opioids. We also have 25 million people who have significant daily pain, for whom opioids have kind of been their only option, although a terrible option. And we need to speed up the process of finding other alternatives. And that is also something biomedical research can do, because there's a lot of really interesting neuroscience in this space. Over there on the right of your picture is actually the structure of the mu-opioid receptor. That's the receptor that all these opioids bind to, and we now know it in great detail. Here's an interesting potential pathway, and this brings us back to genetics. There are people, rarely, who are born with complete congenital insensitivity to pain. They cannot feel pain. Now, you might think that would be a blessing, but it's actually a curse, because those individuals don't know when they've been injured, and they often end up getting drawn into circumstances where they do terrible things to their bodies, like putting a knife through their forearm just to show people how incredibly courageous they are. Even though they don't feel the pain, they end up in sideshows and they die an early death. What's causing their insensitivity? It's this sodium channel, NaV 1.7, which is a critical sodium channel for pain to be transported up through the peripheral system to the spinal cord and above. So think about that. If you could make an antagonist to NaV 1.7 that somebody could take temporarily, that would be a very powerful analgesic, but it in no way interacts with the opioid system, so it should not be addictive. So a lot of work in the basic science, and now moving into the applications through a few companies to get that to happen. Even we might be able to bring the immune system to bear. This always surprises people. What? You're going to make a vaccine for opioids? Well, why not? If you could, okay, those are opioids. There's the brain. If you could keep the opioids from getting into the brain, which they do all too readily because they cross the blood-brain barrier, then you could protect somebody from addiction, because it would do them no benefit to actually go out and find an opioid. And this is critical for people who are trying to stay clean and avoid a relapse, and relapse rates are very high. So, okay, let's make an antibody that actually detects heroin or fentanyl or one of the other opioids, and let's put that into the blood. And then when the opioids appear, they get bound by the antibodies, and in that state, they can't cross the blood-brain barrier. And so you end up greatly muting or maybe completely abolishing the effect. We're actively working on that between NIDA and NIAID. And while it is certainly a high-risk approach, it might turn out to be pretty useful. Some of these antibodies have half-lives as long as 6 months, so you could do this in a preventive way. We need those strategies, but we need not to abandon the people who have pain, so we have to push on that as well. Then I got to get cancer, because cancer is in such an amazing place right now. and particularly the advances that have just burst forward in the last few years in immunotherapy. And some people think that came out of nowhere. Let me tell you where it came out of. It came out of the clinical center over there, and particularly Steve Rosenberg, who's been working on cancer immunotherapy for decades and having failure after failure after failure. Steve would tell you 65 patients where nothing worked, and then the 66th one, there was a response, initially working with melanoma and now extending that. And now with the realization that cancers do have the ability somehow to shut down the immune system's recognition of those cancer cells, and that you could wake the immune system up again with checkpoint inhibitors or with CAR T cells, as in this diagram, where you basically educate those immune cells to know what to go after, kind of taking them to school, telling them what they've been missing and that they need to pay attention to. And the results of this, nothing short of dramatic. Always happy to show this picture of Emily Whitehead, who was the first patient treated with CAR T cells at the University of Pennsylvania by Carl June. She had ALL, which sounds like, oh, that's not so bad, we cure those. Well, we didn't cure her. She went through the standard chemotherapy and more after that, and still relapsed repeatedly, and was in desperately sick shape, and was treated by Carl June with CAR T cells. That picture of her is, I think, from a couple months ago. She is now 5 years out. She didn't just have a response, she's cured. I'm willing to say cured after 5 years with no evidence of disease. Now, in the next 10 years, we're going to see a lot of advances here. The big challenge, and the one that I think everybody is now working hard at, is how do we get this to work for solid tumors? Because so far, for pancreatic cancer, for prostate cancer, for breast cancer, for glioblastoma in the brain, we haven't had nearly the kind of success that you would hope for. But yet those tumors also are making abnormal protein antigens that should be recognizable by the immune system. So what are we missing here? How do we take this to the next level? We've taken the immune system to high school. Maybe we could take it to graduate school here and figure out how to recognize those. I put up here this one paper from Steve Rosenberg from 2014, where in a specific example of a woman with cholangiocarcinoma, a gallbladder cancer, he was able to get this to work, but it was incredibly challenging by sorting out the specific CD4 positive T cells that were like 1 in a million, and then expanding those by knowing exactly what was going on in her cancer in terms of what antigens were there, and finding this rare cell that should be able to respond. That doesn't scale very well, but it's a proof of principle. And I think one of the things we're trying to do with this new collaborative effort called PACT, which is the Partnership for Accelerating Cancer Therapies, which just got started a few months ago. This is NIH working with 12 biopharmaceutical companies, is to try to identify, why does immunotherapy work when it works, and why does it not work when it doesn't work? If we understood that, and we need biomarkers to do that, then I think we could make some real progress. So that's going to be big. Well, number 10, I have to finish my roll call here of 10 for 10 by telling you about a project just launched 2 days ago. And this is a precision medicine project called All of Us. This aims to try to provide a platform for understanding not just how to treat people who are sick, but how to keep people healthy by initiating a cohort study, the largest ever undertaken in this country, of 1 million people, inviting them to come and take part in a study where they will, as our partners, help design the study, make their electronic health records available, have their blood drawn, and ultimately subjected to complete genome sequencing, fill out a whole lot of questionnaires about their health behaviors, their lifestyle, their diet, walk around with wearable sensors that keep track of what's happening to them, and report regularly back on their medical experiences. And a very diverse population. This is going to be at least 50% traditionally underrepresented groups, African Americans, Latinos, Native Americans. We're taking advantage of increased interest in patient partnerships. Now, people want to be involved in this if they're really being taken as partners and not as subjects, and they want their data back, and we're going to give it back to them. We have electronic health records. Yeah, they're clunky, but you can use them for research. We have technologies, particularly wearable sensors, and the omics part of this is getting faster, better, and cheaper. Of course, this is a huge data science challenge, but one that we think we're up to. So this program, which again, we've been beta testing for the last year, And even the beta test enrolled 42,000 people, which makes it one of the biggest studies that NIH has ever done. That was just the pilot. The real launch was actually on Sunday across the nation with various of us scattered around amongst different parts of the country, interacting with communities about this. And we want you to join this too. And you can, because this is open to everybody. I was on Sunday at the Abyssinian Baptist Church in Harlem, in New York City, a remarkable landmark, together with Dara Richardson-Heron, who you see on the left, who's the Chief Engagement Officer for all of us, and Eric Dishman on the right, who was in Detroit. And we were all linked up by a real-time satellite link, doing this as a simulcast from not just those two places, but 5 others. So there were 7 places around the country, all joined together in real time to try to start this enterprise and get the buzz going. And I think it succeeded. We've been a little overwhelmed by people coming forward in the last 2 days, wanting to know more information coming onto the internet site. You might have seen or might not have yesterday in USA Today, this editorial by myself and Secretary Azar, encouraging people to sign up and be part of this national adventure. And yeah, all you got to do is go to joinallofus.org, join all of us, one word, no spaces.org. And if you're interested in finding out, it'll walk you through a lot of educational stuff so you can decide whether this sounds like something you want to take part in. It's open to anybody over 18. We're not doing kids yet, but we will by next year. And again, we're particularly interested in diversity. And after you go through a number of the educational parts, if you're still interested, there's a consent form. And everybody who's going to be involved in this will be asked to consent. And the consent says you're joining on not just as a one-shot deal, but basically in an understanding that this is a longitudinal study that will go on for at least 10 years, and I think it'll probably go longer, and that we will be back in touch with you regularly. And we are going to give you information about yourself as we learn it. And we're also going to ask you occasionally if you're interested in taking part in some follow-up study that might not be done on all million people, but might be appropriate for you to consider, because this is going to be a platform for clinical research of all kinds. So this is a game changer, and one that I think we're all pretty excited about. So that was my number 10. Oh yeah, that's me getting my blood drawn, because I signed up and had my intake done at the Harlem Hospital just Sunday morning. And yes, I agreed to all of that. I consented. I gave them access to my electronic health record, which is really crummy because I don't have a particularly good system for my current doc. But hopefully some useful things will come out of this. And it didn't seem right to go and talk to all these people about what a great idea this was if I hadn't already decided to become part. And I'm honored to be able to do so. Well, that's where we are. I started out talking about the National Institutes of Hope. And just to finish that thought, hope is a word that people throw around a lot. Oh, I hope you feel better, or my hope is that tomorrow it won't rain. I think for what we do at NIH, hope has to be attached to more than just a nice idea. It's got to be attached to action. That's why I like this quote. Hope in every sphere of life is a privilege that attaches to action. No action, no hope. Well, that's why we're the National Institutes of Hope, because we're all about the idea of action, about taking this amazing scientific revolution that we're in the middle of and bringing that forward from the basic to the translational to the clinical, and ultimately to answer the needs of those people whose hope still rests in what we might be able to bring to them. And the next 10 years are going to be unprecedented, I think, in terms of the ways in which we're able to answer those hopes. But we should not take lightly just how challenging this is going to be and how much work we have to do, which is to say, sort of final comment then, the most critical resource we have for success going forward is not all those fancy instruments that I might have talked about. It's not the fancy buildings that some universities occupy or that we even occupy here. It's the people. It's you guys. It's all of you who are going to be the ones who innovate, who think that bright idea that nobody thought before, and who figures out how to bring that forward, who are willing to take a risk on a project that maybe has 1 chance in 5 of succeeding, but if it succeeds, it's going to change everything. That's our most important future hope, in that case, is your action. So I will stop there and see if you have any questions. Thank you all. And yes, the gun is still here in case you're worried about that part. Questions? And there's a microphone there. Yes, please.
Thank you, Dr. Collins. That was a great presentation about all different areas. But I wonder if you can talk a little bit about the larger scale, about the mind-body connection, about environment, about social influence on health.
I'm glad you raised it because I think that's a critical part of really understanding health and also on the onset of illness and how best to manage it. When you look at the opioid crisis, for instance, we will not succeed in understanding or turning this around unless we understand the social circumstances that create the unfortunate circumstance for people both to fall into this trap and then to have trouble getting out of it. We probably need to figure out better ways to treat pain that don't involve drugs and don't involve devices. Perhaps the things more along the lines of meditation, cognitive behavioral therapy. When you look at some of the data that's there, in terms of the value of those, it's pretty darn impressive, even though a lot of the sort of hard science people will immediately be skeptical. Look at the data. It's really quite impressive. And certainly with all of us, one of our goals is to have that be a truly holistic view at what are the factors that play out in terms of health and illness. And that includes social stresses. We aim to have a heavy representation in that group of people in the lower socioeconomic groups whose lives are very different than all of ours, to understand what that influence is. If anybody thinks that's a genomics project, genomics is the easiest part of all of us. The really difficult and probably more important part is the environment and the social and behavioral situation. So thanks for raising that. I know people always think, oh, it's Collins, he only thinks about DNA. And yes, I do think about DNA, but I think more broadly about it in this context. We have to put it all together. Yeah, other questions? Here's one.
I was wondering, with 27 institutes and centers, do you ever, just like on a nice day like today, walk along and dream, what if there weren't centers that were devoted to specific diseases like the Cancer Institute, or ones devoted to body parts like the Eye Institute, and you totally reorganized how NIH was set up? Do we ever think about that?
Yeah. You know, after he left as director, Harold Varmus wrote a paper about that, suggesting that actually NIH would be better off if it was reorganized into just 6 institutes with some thematic areas, but not so much. It was a bit controversial. I think if one could just basically wipe the slate and start over, we probably would not organize NIH the way that it is. But we have learned how to work with that structure because we've also learned through advocates and through the Congress that messing with it doesn't have a whole lot of chance of changing things. So we better make our function behave as if we didn't have all those different components. And I think that's what we've done pretty effectively because the institute directors believe in that as well. You know, we tried 4 years ago to do the simplest of these, which is to merge the Drug Abuse Institute and the Alcohol Abuse Institute. I mean, doesn't that sound like a place you could start? And we spent a year and a half putting together all the pieces and then basically got told by the Congress, no, you don't. So if we couldn't do that, I don't think we can do Harold's view of something much more dramatic. So our challenge then is to figure out how to work within that. There is this effort right now called Reimagine NIH or Optimize NIH, which Larry Tabak is leading. And we are identifying areas of an administrative sort where having 27 of everything is probably not necessary, and we could be more effective if we figured out how to work together in some of those functions. But that's a modest sort of step in the direction of trying to work within our constraints, but I think it's a good one. Scientifically, things now have gotten so interdisciplinary that the idea that any institute would succeed if they decided they were going to do everything themselves, they would quickly fall behind, and I think they know that. Yeah, right here.
Thank you so much for that lecture. It was great. I've worked in the House of Hope for the last 8 years, and I just agree with what you said about just being in an amazing place at an amazing time. And I spent a lot of time thinking about some of the concepts that you covered when talking about your early career. So you talked about competition, going from competition to collaboration, and making, data sharing and just working sort of not in silos. So, and this is a general question that I'm sure you've received many times, but I know that there are a lot of trainees in the room.
Yes.
So I want to know either what's the best advice you can offer us as intramural investigators, and or the most important skill that you believe is important for early career scientists or clinicians to develop?
Yeah, I have been asked that, and I always worry that people might take me seriously. So let me see here. As I said a little earlier, I think one thing, particularly for early stage investigators, is if you're the one who's now going to decide what you're going to work on, right? If you're going to pick a project, try to pick something that's significant if it happens to work, even if it's a little risky. You're going to put a lot of your time and effort into this. Why not choose a project that has some fundamental aspects to it, whether it's basic or clinical, that if it succeeds, it's more than just, a paper that everybody will forget in a couple of years. It's actually adding to the body of knowledge in a way that is going to change things. So that would be one thing. Pick areas that you think are going to be substantial. Certainly when it comes to data, we have to all get past what were sort of the bad old days where everybody held on to their data for much too long. And that does nothing to benefit the enterprise, and it does nothing to benefit the investigator. I've never seen an experience really where an investigator who was free to share was punished for it in terms of the way in which they were viewed by the community. And I've seen plenty of instances where those who hoarded lost out as a result, even though maybe in their view they thought they were just protecting their own career. In the long run, it doesn't work. So being open, and of course, being willing to collaborate. I told that story about cystic fibrosis. My lab wouldn't have found that gene by ourselves. Lab Chi Choi wouldn't have found that gene by themselves. When we put it together, we got there probably years sooner than would have happened otherwise. That idea of identifying whatever you're working on, what's a skill set that would really help, but you don't happen to have? Well, go out and find somebody with that skill set and say, okay, let's do this together. Stuff happens so fast when you get in that circumstance. And yet it's remarkable how people are sometimes reluctant to get outside of their own comfort zone. Up here in front.
Thank you, Dr. Collins, for a great talk. My question is more related to the last number 10 that you showed about precision medicine.
Yeah.
So the question I have is, in the literature, there is discussion about personalized medicine and precision medicine. And then if you could kind of comment on the distinction as well as the confluence of the two, which you see with the other 9 breakthroughs or areas that you discussed.
They are all intersecting. I'm glad you brought that up, because by putting forward 10 ideas, you might have thought, well, okay, we could pursue those somehow separately from each other and all would go well. It should be pretty clear. If we're going to understand the brain, our brains are all different, so there has to be an individualized idea about that. If we're going to study single cells, well, they all have a genome and they have a variation in that genome, so you can't understand them without considering precision. Cancer immunotherapy, undoubtedly, there are aspects of that which are going to vary from tumor to tumor, from person to person, so it's got to be in there. The distinction between personalized medicine and precision medicine is a little subtle, and I'm not sure I've ever completely bought the need for the switch. But I wrote that book now 9 years ago on personalized medicine. The National Academy of Sciences had a very influential group that talked about where we were going, and they preferred precision medicine because they thought personalized medicine sounded like you were really talking about something that only worked for that one person. Whereas their argument would be, we need more data so that we can see in a very large group of people, well, this group over here of 20 or 25 is similar to each other and different than the rest. So you're doing precision medicine, but it's not just like N equals 1. That's the rough distinction. I tend to blur that a bit. Sometimes I forget and call it individualized medicine because maybe that's on the same spectrum. Yeah.
So if I could take my own example as a person with certain diagnosed diseases or ailments. Or I, as an individual, of course, I am interested in precision medicine, large cohorts, but for a day-to-day life, what I am looking for is what can help me to manage or address my individual problems.
Yep.
So that is what my question was, the dichotomy between the two, one that can benefit a large populace, that is great. But then there is always the individual whose life needs could vary from the others.
And that is the sort of the tension, but also the opportunity. And that's why we need really big numbers. That's why we're talking about 1 million people and not a few thousand. Because in 1 million people, while each person is individual, they're likely to be clusters of people who are more similar. And if we could learn that, okay, in 1 million people, there are 32 of them that have very similar circumstances to yours, and that some of them already have some medical experience about what has worked and what has not worked, then we can offer you something better than the one-size-fits-all approach, which is what we're currently stuck with. It won't be N equals 1 just about you, but it will be based upon people whose similarity to you is substantially greater than we otherwise would have known. Yes, one more, and then I think we'll probably move to this silly conclusion.
So you started your career in chemistry.
Yes.
And so there are theories like quantum mechanics that explain
I love
that discipline very well. My background is in relativity and astrophysics, so there are also good theories in that area. And in biomedical and in biomedicine, we still seem to be struggling to get to a sort of a level of par with chemistry or physics in terms of how theories can explain nature, and in our case, life, of course. Where on your scales do you see we will achieve some kind of advancement there? Is that may not be 10 years out, but maybe you can have a projection how we're progressing there?
Yeah, the complexity of biology, which of course builds upon the fundamentals of physics and chemistry. That's, if you want to ask, what's really there, it's physics and chemistry, but it's in this complex system. But there are still some fundamentals about biology. Certainly the whole idea of information content by DNA and how that plays out in terms of determining the phenotype of a particular cell by which genes are on or off. Certainly what we've learned about genetic variation and its influence on outcome. I don't know if you could call those laws in the sense that Maxwell's equations describe electromagnetism, but they are fundamentals about all living things that you can build a lot upon. And I think of theory as trying to provide you with fundamentals upon which you can build experiments and then test the theory. I think the brain is going to be really interesting in this regard, because I don't really think we understand the first thing about how the brain works. We don't know how you lay down a memory and retrieve it. We don't know how you can carry out complex functions using circuits in your brain without actually putting a whole lot of mental effort into it. It just happens. There's got to be things there that will surprise us. My goodness, when you see some of the images that are coming out of the connectome, which is looking at a particular way of looking at the wiring diagram of the brain, it's breathtaking. All these right angles where things are set up in a way that looks pretty clever in the terms of the engineering. But we're like, trying to take apart a computer by, breaking into it and studying the transistors by poking at them. We don't really understand how the thing works, but we're going to figure it out. And there will be some fundamentals there that have been out of reach, and they're about to be in reach, at least in a 10-year period. Well, maybe we should finish this off, or finish you off, with a small bit of silly. And yeah, at moments like this, I'm prone to trying to come up with a song that might be known by the person I'm singing about. So because Wynn is a little bit past age 35, this song is one from about 50 years ago that I hope maybe he has heard before and will, from this point on, probably never want to hear again. And it's been much manipulated in the lyrical department, mostly by Chris Wanjek, who agreed to be my partner in this. Is the amp still on? Yes, okay, thank you. I couldn't tell from here. And so, yeah, this is a little bit of a ditty about being asked to do this and wondering about whether I could and whether I should, and then finally deciding to. And it's got a little bit of a repeated line at the end of each verse that calls out for participation. And I happen to know a few of you know how to do that, since some of my band members are here in the third row. You know who you are. Being director, I couldn't resist. I was thinking about what I might have missed. When asked me to teach, of course I said yes, but then I had to figure out what things to address. But oh, no one's sweeter than win. Sweeter than wine? Oh, no one's sweeter than win. You didn't know that was coming, did you? I have to get the tune right. It would help, wouldn't it? Now, the course he leads, I'll give you the rap. It's all about bridging the gap between the bench folks with their Ph.D.s and the bedside docs with their fancy M.D.s. Oh, no one's sweeter than win. Oh, no one's sweeter than Win. Win's worked mighty hard, and so has his wife. And this course he's taught, it's taken on a life. Folks learn of advances in biology and their stunning application to human disease. Oh, no one's sweeter than Win. Oh, come on, y'all. Oh, no one's sweeter than Win. Well, Win's worked mighty hard, and so has his wife. And this course he taught, it's taken on a life. Folks learn of advances in biology and their stunning application to human disease. Well, the years have rolled by, and word's gotten round. Demystifying medicine is the talk of the town. And for Win, no speaker can be out of reach, with IC directors all wanting to teach. Oh, no one's sweeter than Win. Oh, no one's sweeter than Win. Well, life's really crazy, you probably know, but Win was convincing a few months ago. Finding time in my schedule brought a whole lot of pain, but oh Lord, I'd do it again. Oh, no one's sweeter than Win. Oh, no one's sweeter than Win. No one.
My grandchildren, thank you. But wait a minute.
Oh no, we're not done.
I wish to give you the award as the #1 bridge builder.
Oh well. May I?
Yes. by all means. Well, needless to say, we all here and those listening and watching and yet to watch.
Oh, a picture history of the Brooklyn Bridge.
Thank you so much. Well, we'll see you next year.